The topic discussed under pharmacodynamics is drug administration and drug action, basically drug receptor interactions and theories.
Drug administration is the process of giving a drug to the patient to cure or prevent a disease or complication. The choice of drug administration depends upon the characteristics of the drug, like its solubility, pH of action, and its bioavailability if taken through different routes. It also depends upon the state of the patient, like age, condition, or stage of disease, dosage, other health parameters, site of desired action, effect of digestive juices, and urgency of response. The different ways of administering drug is enteral, parenteral, and topical. No method or route is perfect. They have their advantages and disadvantages. The choice of administration depends upon the specific purpose, bioavailability, and urgency. Enteral method. This way of administration is done for tablets, syrups, capsules, powders, and granules. In buccal administration, the drug is placed between the gum and the inner lining of the cheek, while in sublingual, the drug is kept below the tongue. In this method, the drug absorption is quick, can be taken by the patient, economic, and first pass is avoided. The disadvantage is that some drugs are unpalatable and cannot be given in large quantities. It may also cause irritation of mucosa membrane. Oral method. In this, the drug is swallowed. It is the easiest way to consume and can be done by the patients. The oral route produces a systemic effect. The drug is absorbed in the gastrointestinal tract. The disadvantage is the first pass effect needs to be checked for the bioavailability. In case of rectal route, the drug is administered rectally. It can serve as a local or systemic route of administration of the drug. Generally, it is given for children and patients with vomiting and post-operative. Advantage is little or no first pass effect. Used can be used in unconscious patients and patients with vomiting. The main disadvantage is absorption is low, inconvenient, and irritation and inflammation of the rectal mucus. Parenteral method of administration of drug is done by injection or infusion. It could be intravenous, intramuscular, subcutaneous, intradermal, intraperitoneal, or intrathecal. The figure here shows parenteral administration of drug by mode of injection. Topical medication. It is a method of application of drug directly onto the surface of the skin or mucous membrane. It could be in the form of lotion, oil, ointments, cream, emulsion, powder, or paste. It lacks systemic reaction. It is only used for local effects. The pharmacodynamic principle. In our last session, different drug receptor interactions were discussed. The binding of the drug to the receptor is the first step. It then activates the effector mechanism to give cellular response. The effector may be the part of the receptor or a separate molecule. The possible ways that the drug receptor binding results in response is represented below. Drug action. A drug can act as an agonist or antagonist. A agonist drug binds to the receptor and produces a response or effect similar to the physiological signal molecule. Its intrinsic activity equals 1. The antagonist drug prevents the action of the agonist. Its intrinsic activity is 0. Partial agonist. It cannot produce the same maximal response as an agonist. Its intrinsic activity ranges from 0 to 1. The binding of the partial agonist prevents the action of other agonists and may thus appear as an antagonist in the reaction. Inverse agonist binds to a receptor to produce a response that is opposite to the corresponding agonist. Intrinsic activity of a drug indicates the ability of the drug receptor binding to initiate downstream reaction leading to effect or response. The graph on the left represents the effect in percentage of different doses of drug. A full agonist drug has similar response to a physiological signal molecule. Hence, its effect is 100%. Thus, the intrinsic activity is 1. Similar, Similarly, for partial agonist, the effect varies from 0 to 100%. Hence, the intrinsic activity is between 0 and 1. For an antagonist, the intrinsic activity is 0, while for an inverse agonist, the intrinsic activity is in negative. There are different theories put forward to explain the interaction between the drug molecule and the receptor. First, the occupational theory by Clark in 1926. According to this theory, the binding of the drug activates the receptor to give the biological response. The intensity of response is directly proportional to the number of receptors occupied by the drug. A maximum response occurs when all the receptors are occupied. The response ceases when the complex dissociates. The strength of the stimulus in the drug receptor complex is affected by whether the drug is an agonist or antagonist. Depending upon that, the DR complex may be strong or weak. Next, the induced fit theory model. This model has been discussed in the earlier session. In this model, the agonist induces conformational changes in the receptor leading to a response, while an antagonist does not induce any conformational changes, so no response. Next, Patent's rate theory. According to this theory, the response of drug-receptor interaction is proportional to the rate of association and dissociation of the drug from the receptor. Next, the two-state receptor model. It is based on the kinetics of competitive and allosteric inhibition. According to this theory, the receptor exists in two interchangeable states in equilibrium, the active, that is Ra, and the inactive Ri. The binding of the drug to the receptor alters the equilibrium and correspondingly the response is made. If a Agonist binds to the receptor, the equilibrium is shifted towards Ra. This leads to positive response. In case of a competitive agonist, this binds to both the Ra and Ri state with equal efficiency, thereby maintaining the equilibrium, hence zero efficiency. A partial agonist has a slightly more affinity for the Ra state, hence submaximal response. The inverse agonist has high affinity to the Ri state, hence the equilibrium is shifted towards Ri, leading to negative efficacy. The drug permeation into the cell can occur in different ways. It could be by passive diffusion, by carrier methods, or by pinocytosis. Aqueous diffusion. This is a passive method of diffusion that occurs within interstitial space, epithelial tight junctions, and across endothelial blood vessel linings through aqueous pores. It is based on the Fick's law. Certain drugs which are bound to the plasma protein, example albumin, cannot permeate through the aqueous pores. Drugs that are charged or charged drugs are also influenced by the electric field potential. The Fick's law describes the passive movement of molecules down its concentration gradient. The flux of the molecules per unit time is calculated by the method or formula given below. Lipid diffusion. For drug permeation, the molecule needs to move across lipid barriers. The lipid aqueous partition coefficient of a drug determines the movement of the drug between the aqueous and the lipid media. Next, permeation of drugs by means of special carriers. Peptides, amino acids, and glucose enter cell through special carrier mechanisms. This could be by means of active transport or facilitated diffusion. The figure here shows the different ways the drug permeates into the system. The table here represents the comparison between passive diffusion, facilitated diffusion, and active transport. Permeation of drugs by endocytosis and exocytosis. Entry of large substances and molecules like iron and vitamin 12 complexed with the binding protein enters the cells by method of endocytosis, while neurotransmission occurs through exocytosis. Thank you.